The overall goal of this microfluidic experiment is to evaluate the effects of kinase inhibitors on tumor cell migration under a direct electric field.
So this method can help answer key questions in cellular electrotaxis studies, such as what are the determinant pathways in electric field-guided migration of cancer cells. So the main advantage of this technique is that it is a highly reproducible and time-saving electrotaxis study that can be easily implemented.
The implication of this technique extends toward the diagnosis of metastatic potency of cancer, as the mobility of malignant cancer cells can be enhanced by an electric field.
So this method can provide the insight into the electrotaxis of tumor cells. It can also be applied to skin regeneration processes, such as wound healing.
Begin by using a drafting and design program to draw an individual acrylic layer pattern. Save the pattern, and then turn on the laser scriber. Connect the scriber to the controlling laptop, and click on Designed Pattern File to open the acrylic layer pattern. Next, place a piece of blank acrylic sheet on the XYZ stage of the laser scriber, and use the appropriate corresponding laser scriber auto-alignment stick to set the focus of the laser beam on the surface of the sheet. Send the designed pattern to the laser scriber for direct machining of the acrylic sheet. Then use forceps to remove the protective paper from the acrylic sheets and blow the surfaces clean with nitrogen gas.